And let folks who are coming in catch up. We're going into oral session 3, and we have 2 speakers in this session. Our first speaker is going to be Dr. Lakshmi P. Kotra, and Dr. Kotra works in the area of medicinal chemistry, preclinical and clinical development of small molecule and natural product-based drugs. He is the recipient of many awards, including the Premier's Research Excellence Awards from the province of Ontario, R&D, Health Research Foundation, Research Career Awards, GSK, and many others. Dr. Kotra is going to speak to us about bench to patient to bench is always crunch time. Dr. Kotra.
Thank you very much. First of all, I appreciate the opportunity to be here at TGHRI Research Day and share some of our research that we've been carrying out at UHN. Probably now this is my 13th year going forward. What I mean by the title, when I was asked to give a title, I wasn't sure what to give, so I gave the title that you see. But what I really mean is that we work in small molecules as drugs that you see on the left-hand side, and with the idea of moving into the patient and back and forth. And you're going to see that resonated as I take you through the slides that you're going to see this afternoon. And these are some of my disclosures. I do have some conflicts with respect to private industry, and whatever I'm saying here is purely my own opinions and thoughts, if you will. We've been very fortunate over the years to receive funding from a variety of sources, both national and international sources, that supported our activities. And I don't want to forget acknowledging our fantastic support teams we've got at UHN, as you see, the animal resources, facilities, tech transfer, and our RIS folks. And I know this for a fact because I know without their help, I could not have mounted what we've done with the efficiency that we have been able to accomplish some of the things. And these are all the talented people on the left 3 rows that I work with or continue to work with. Those in the bold are in my current group. And on the right-hand side column are all my collaborators, both in Toronto, Canada, and elsewhere in the world. So, drug discovery. That is probably the punchline if you saw the abstract that's in the book. And usually, I'm just going to take 1 minute here to illustrate that. When we talk about drug discovery, it means different things for different people, depending on their background. To me, what it means is that a molecule, a substance, let's put it that way, that is useful for a patient, useful for a healthcare practitioner to be able to use it on a patient. That's what it means for us. Thank you. So these are some of the programs that we've been fortunate to be associated, mainly led by our group. And what I'm going to do today is to take you through some of this. I will give you some milestone slides, and I will show you some fun slides. But at the end of the day, you will see the big picture that I put out in my abstract of the tenacity, persistence, opportunity, support of the village, and words of that nature, how they impact us. Okay, so then on the top you see anti-malarial agents, ODCAs. That's one of my first projects I started in Toronto when I moved from US. And I'm going to give you some developments on that. On the antivirals and interferon mimetics, we collaborated with Eleanor Fish, and we haven't been able to move past the early lead stage on that. And with thrombocytopenia with CBS, Don Branch, multiple sclerosis with sick kids, and diabetic neuropathy, and very recently on medical cannabis. And my topics today I'm going to cover would be on the anti-malarials to antivirals shift and the development on that, if time permits, multiple sclerosis, and a couple of slides on diabetic neuropathy, followed by the topic of the day in Canada, probably in the world, on cannabis. All right, ODCAs. It is basically rotadine monophosphate decarboxylase. This was originally started as a business deal between me and Professor Emil Pai, who just retired this July. And I was at the time looking for support for a modeling center, and he was looking for someone to work on inhibitors for his enzyme, which is that enzyme. He knew at the time that I was trained by the gentleman that you see there, Professor David Chu, one of the legends in nucleoside drugs. If you know Harvoni, which is a Hep C drug, he is the guy behind that, and many other HBV and HIV drugs as well. So Emil and I were able to bond very quickly. This is a biochemistry 101, how nucleotides are synthesized from amino acids, de novo biosynthesis. If we delve into that a little bit more, you see the last step at the bottom of the screen, a molecule being synthesized, that's called UMP, which all of us understand from Biochem 101. This particular enzyme I talked about makes that particular UMP molecule in the cell, which means that it's pretty much present in every cell except viruses. In some cases, it is essential in parasites and things like that. There are redundant pathways in higher species. That's a dimeric structure of ODCAs on the left-hand side that's turning around. And the actual chemical reaction that you see is exactly the carboxyl group that's down there. It's decarboxylated to make UMP. You see talented people in my group, Angelica, Eva, and William contributed quite a bit in advancing this particular project for us. This is all the chemistry that we've done over the years. You see some of the published papers on that, so there's a lot of chemistry that goes on in our laboratory. The idea of all this chemistry is to manipulate each and every functional group or atom in these molecules to see what's happening, how the enzyme responds to it. Simultaneously, we filed patents on this very early on. Tom Parsons is sitting here. He helped with the filing of this. And the UHN holds patents on these things. And as a PI in my early days, as interested in drug development, had to deal with it. And I'm going to show you 3 slides, some exciting science that came out of it. This is one of the inhibitors we synthesized, and I'd like to draw your attention to this little guy here that all of us know, C triple bond nitrogen, so there are 2 atoms there. When we give it to the enzyme and do the crystallization, it comes out as only 1 atom. You see that just 1 atom stays put there. What happened is that the 2-atom nitrile group transformed into carboxyl, the hydroxyl moiety. Even today, we have no answer how the enzyme does it. There are a lot of groups that followed upon our work trying to understand the chemistry, how the enzyme does it. It cannot happen without the enzyme. It's impossible to happen from the energetics point of view. So that was an exciting publication and the work that came out of that. The other work is, you see the little surface on the right-hand side at the bottom, that's enzyme active site. And I'll give you a little picture of how important it is. Think of uridine monophosphate. It's a precursor to cytidine and thymidine. And a cell is replicating fast. It could be human cell, could be parasite, could be virus. HIV doubles its cycle every 20 minutes. UMP is essential as one of the pyrimidine nucleotides for all other things. Means that this enzyme, if it doesn't work fast in milliseconds, you don't have the pyrimidine pool that you need. So, which means that the active site is extremely conserved, very efficiently evolved. That's the surface that you're seeing here. Now, turning the tables a little bit here, uridine, as you see here, 6-member ring, right? And you see all these other rings that you see, 2 rings, 1 ring with all other substitutions. For the first time, we've shown that this enzyme can accept a variety of substitutions on it and can actually be inhibited by this. This active site is not as rigid as once people thought. And by the way, this enzyme is known for about 40 years. This is another first. People never thought that this enzyme could be functioning as a covalent inhibitor. We discovered a covalent inhibitor for the first time. A very efficient enzyme and the covalent structure that you see through X-ray crystallography and things of that nature. Okay. At the time, we learned about a very young and very bright parasitologist at UHN. I was at U of T at the time, Dr. Kevin Kane, and we started collaborating with Kevin and Ian Crandall and evaluated our compounds for malaria. And the molecule that you just saw, the covalent inhibitor, showed very good activity, including in drug-resistant strains. And fast forward 3 years, we were very fortunate to develop a collaboration with the Indian group. There was an India-Canada consortium that got created. We got funding from both governments in developing these compounds. And you see the teams, the Indian team down here and the Canadian team. We had industry as well as academic groups, multiple groups participating in this to advance the anti-malarial that was discovered in the tissue at the in vitro stage into the in vivo and follow-up development. So you're going to see some slides that go beyond activity finding, what is involved, and this is all the types of things that we have to do as an academic group, without which the drug would not really go forward, a molecule would not go forward, and it just dead. So in that context, here you see some solubility data. This is a very well-soluble molecule that you see down here, 30 milligrams per ml. That's very good from the drug standard point of view. And something called polymorphism. What this means is that if you have the compound, how does it change its physical structures? Think of similar to sugar crystals, if you will. If it changes into different crystal structures, it cannot be a good drug whatsoever. And we've shown that it does not. It retains its properties. And this is a combination drug therapy that we were doing. And this varies from every disease focus we have. In cancer, in antibacterials, in antimalarials, combination drugs are essential. So if you want to do drug development, you got to put your head into that. On the left-hand side, you see control mice. There's a very aggressive malaria. Day 3, day 4, by day 7, mice die. It's P. Bergii infection in a mouse. In the middle row, you see a 3-drug combo. On the right-hand side, a 2-drug combo. And 2-drug combo does not function very well by day 7, just like controls. All mice die by day 6. And the 3-drug combo, day 7, they're good. And by day 11, it's all cleared. Now mice are doing really well. So we've been able to show that drug combinations work as well. And that's very important at the preclinical level before one goes into the clinical studies in this context. Along with that, drug formulations, you see a couple of tablets there, 300 milligram dosage tablets, which is from the human standpoint of view, that's the right dosage, and other tests that need to happen on those tablets. Are those good tablets, bad tablets, things of that nature, in addition to everything else. And so as I said, Emil Pai, that's how we got started with, and after a few years, Emil said, Lakshmi, I'm now done with this drug development. I leave it with you and give me an update every year what's going on. So he is out of the picture, and we are still continuing this with the Indian team. Animal efficacy studies, toxicology, those are pretty much complete. We are currently at the stage of preclinical package preparation for the submission to Indian government. That's 2018. So the project essentially dragged along, for a lack of a better word, for about 13 years since the discovery of the ODCAs that we started with. So it does take a long time. Now, you heard a lot about PhD discoveries, the impact, science publications, right, so far. Those of you who are still young, who are looking at going into future careers, whatnot, things like that, and have any aspirations of drug development, timelines, right? And, of course, the money that needs to go into all of that. That's another thing to cap on that. Okay, so the little structure there, and you see the 2-letter things, Copacamal, right? Kotra pi caine malaria drug. That's how the name came up. Okay, and then about 2 years ago, we met Dr. Atul Humar. He's at MOT at TGH, who was interested in antiviral drug, especially drug-resistant viral infections that are reasonably common in the organ transplant. We were able to pivot very well to move our ODCase inhibitors into that program. And last year, we screened our compounds in that there is some mediocre activity, not excellent activity. And currently, we are in the process of refining. And you see on the right-hand side, these are CMV plaques. Virus only, you see cytopathology, essentially pretty bad there. A couple of our compounds showing some recovery from that. Now we need to optimize that. So we're in the process of doing that. Adam and Daniel are working on that, pushing that forward. So that's a pivot that you saw, antimalarials, antivirals, so both of them are going on. Now I'm going to talk to you in about 4 slides a program that I think we got lucked out quite well, and this is where the luck plays a role, if you will, in that, which is the diabetic neuropathy. This originally started with JDRF, where Paul Fernahue, he's in Manitoba, and Nigel Calicut were trying to develop drugs that would cure diabetic neuropathy. JDRF said you need a medicinal chemist, so go talk to Kotra and get him into your grant. We'll fund you. So we got that funding for that, and you see the expertise that's in there. What's the idea behind diabetic neuropathy? What they're going after? There are a variety of neuropathies. In this particular case, the small fibers that are at the tips of the sensory neurons and all, they degenerate. And the worst case scenario, all of them basically degenerate to the dorsal ganglia and things of that nature. So you don't feel anything, things of that nature, which is pretty common in diabetic neuropathy. We wanted to look for those that would reestablish that, not only prevent that degeneration, but regenerate all of that. On the left-hand side control, you see the neuronal cells, pretty very weak fibers that's showing up, preventing, reversing neuropathy. Then now you can see all lighted up after the treatment with the drug. There is a screening system that's established and patented on that, and full nerve growth established in vivo models being shown up as well. It took shape very quickly. There's US, Canada, all three of us were very interested in developing it forward. And there's a three-way company that got started in San Diego, Vincentor, and to cure diabetic neuropathy. That's the patent that three universities hold it together, and an executive team that's moving that forward as well. So you see that now Vincentor, Winnipeg, San Diego, Toronto. That's how the name came out there. So these are the fun things that one gets to have as things go forward and they go out of the academic lab into the private sector, and one must be prepared for that. Okay, I have about 30 minutes. So I'm going to skip multiple sclerosis, maybe 2 slides, but then move on to medical cannabis on that. This is a project that came to us through Mars Innovations about 8 years ago. A neurologist by name Dr. Mario Mascarella, he passed away in 2013, who spent about 40 years looking at peptidyl arginine deaminase. It's an enzyme. It's prominent in the brain, especially in MS tissues. And he understood that very well. And when he approached me, they had something, but they really don't have anything in terms of intervention as a small molecule. And you see the team there. We work with Shannon Dunn at TGH, Peter Strice from Calgary, and my talented team down there. We entered through computer modeling, identified a few molecules, and fast forward, just this year we started a collaboration to move this into drug development mode with CDRD in Vancouver, where we now have a lead compound, and essentially this molecule prevents and reverses neurodegeneration in multiple sclerosis, which automatically leads into the suppression of any immune reactivity, whatnot, things of that nature. So such evidence currently we have from multiple laboratories showing that happening. Okay, topic of the day, medical cannabis and cannabinoids. I've got about 10 more minutes, and I'm going to see what I can do in this. This is a picture that's taken this morning at 12 o'clock and 5 seconds, or 5 seconds after midnight, okay? Nikki and Ian, first legal purchasers of cannabis, not medical cannabis, cannabis, and that's Bruce Linton, the CEO of Canopy, a $20 billion company in Canada. Now, you see the words flashing out there. Exactly a week ago, I got a phone call, and is it University Health Network? Yes, I'd like to do some cannabis tested. You know, I'm reasonably busy in this, so sorry, I can't do that, but who are you? Who am I talking to? I don't get the name from the other guy. Are you really University Network? Can you do the testing? I said, no, tell me who you are, if I can help you or not. I can't talk any more than that, because you get all kinds of calls in this type of thing, right? Then he opens up a little bit. My daughter. And she said she would stop using, and I gathered from his voice he probably is not only concerned father, but his daughter is younger, and he wants to get everything tested. I said, no, we can't help with that in that particular context. So The reason to bring this up for me here is that cases like this hang over in the back of our minds when we take up any projects of this nature, whether it is this or anything like that. But this is, of course, of the topic of the day, and it's always in the back of our mind how these drugs could affect adversely. Do not try this in lab. Those are the cannabis samples we got in our lab. It's all totally regulatory controlled and security controlled. So any of you, if you are thinking about trying to get something in the corner, take it to your lab, don't do that. It violates all kinds of laws. I can talk about it 3 hours, but our collaborators in the cannabis project, Hans Clark, Albert Wong at CAMH, Mamata Bhatt, MOT, Del Campo at Western, our talented team that do everything from chemistry to in vitro, in vivo clinical studies in this aspect. Challenges. Cannabis has a huge challenge right now where we stand here. It's generally considered harmless by public. And by the way, whether we realize it or not, it is not a single substance, cannabis itself. If you go into a store, you will find 300 different varieties of cannabis, which automatically tells us that even the cannabis itself is not single substance, let alone there are hundreds of chemicals inside that cannabis. There is overwhelming anecdotal evidence integrating cannabis into pharmaceutical care. That is what currently we are trying to look at. And the place of cannabis as a medicine in the modern pharmaceutical care. So those are the types of things that we are interested in. That's how the bud looks like. On the right-hand side, you see the famous molecule, tetrahydrocannabinol, or THC. Give or take, there are right now about 500 plus different varieties of cannabis, depending on what part of the country you go to in this case. And tons of licensed producers. These guys have licenses from Health Canada to cultivate and sell, and a variety of chemicals in those. Let's not get into further details on that, but this is a little slide to impress you with the biosynthesis of these cannabinoids in the plant. And more importantly, what I want to tell you is that the genetic variants upregulate, downregulate different enzymes in these plants, hence the concentrations of all of those compounds can be manipulated, and people do manipulate when they produce them. You see 568 chemicals, that includes cannabinoids, about 120 plus in those things. That's how the varieties are now made. So when you see a cannabis, there is a lot of complexity from the chemistry standpoint of view. I'm going to round this up in my last slides, and you'll see how this all plays out at the patient level. Okay, from the body standpoint of view, there are cannabinoid receptors. We have endocannabinoids, or molecules that work on cannabinoid receptors in our own body. CB1 and CB2 are the most common. There are a variety of other receptors as well. I think I have a slide on that. CB1 is primarily associated with the neurotransmission, CB2 with immune regulation. And believe it or not, we have more CB1 receptors at the presynaptic neuron than every other receptor combined, opioid and other types of receptors that we think about. So there's the population of this CB is pretty heavy on the neurons. So there must be a reason evolutionarily for this. And this is a busy slide, but my take-home message out of this, as I just mentioned to you, it is not just CB1, CB2, but these plant-produced molecules, all of these that you see on the left, work on a variety of other receptor systems that are tied into appetite or psychological and a variety of other activities that patients claim to see happen that you're seeing there. So, what am I dealing with if I have cannabis with me? A complex substance that can act in many different ways depending on what exactly is in there. My laboratory has a very wide research interest in this, understanding the plant or the medicine in the plant, its medicinal chemistry, extractions, delivery into the body, that is trying to make cannabis as a medicine, if you will. And also its effects on the genes, cells, receptors, and different animal models, PK, and clinical studies reaching that. In this particular next few slides, our questions were, what's in the plant exactly, chemically? And what's sold to the patients from this? Again, are we finding same thing that we want to find in the plant? and a concept called biological potency that we're trying to advance, where currently the industry really doesn't recognize anything but the THC concentration. Okay, so this is a slide that you're going to see Melissa here trying to extract cannabis extract. It looks pretty much like that. You take the bud, weigh, and you can use multiple procedures to create that extract. And the funny thing is that heat, whether it is 30 degrees, 60 degrees, 100 degrees, 150 degrees, doesn't matter, changes the chemical composition. And the plant produces this guy on the left-hand side with a carboxyl group. Heating, if one is smoking, generates that after you heat. This is a very active molecule, highly potent molecule, binds to the receptors. This guy does not. Same with these two as well, okay? So a practical view here is that I have the plant that I buy or the patient buys. I can make a tea, I can make a cookie, I can make brownie, or I can smoke, or anything in between. What exactly is happening? How much of that left-hand side molecule that you see is getting converted into right-hand side? I bake a cookie, I convert 20%. You bake a cookie, you convert 100% from the same bud. You're going to get 5 times more effect because you got 5 times more compound in your cookie than my cookie, right? So that's the practical side of it. So what Melissa did was that she went in, we got some processes in the lab, in the chemistry, different procedures. Some of those use heat, some of them don't use heat. And try to understand the chemistry of what's coming out. Now, the next 2-3 slides are going to be a little bit chemistry intensive, but here is to communicate a message. So this is something preheating where we did not apply heat at all at 25 degrees extract. This is how the compounds profile looks like. Now, all you need to pay attention to is how these peaks that you see, those lines are shifting when we heat. You see that peaks shifted? There are about 63 chemicals we can track how they shift their chemistry in that, and we can track that on a regular basis. And with that, what William did was that he started to identify each and every one of those molecules that goes beyond the THC CBD. And there are about 196 chemicals that in the sample that we use that includes not only cannabinoids, every other chemical. And that's the natural cannabis that is unheated. And this is after heating. You see the number of peaks changing, number of compounds changing, and there are some common compounds, there are some different compounds. We understand very little about any of those differences, how they are impacting. This is the list of compounds to impress you that it is important to pay attention to that, especially in the medical context. A very recent report about 3 months old out of Oregon that some of the terpenes in these cannabis products, when lit into smoke or when one smokes, generates benzene and other toxic chemicals that usually we frown upon from the health standpoint of view. So the general idea that cannabis is safe is not really true. We just don't have enough data on it. Now, why is this important from the point of view of anything? What is patient consuming? That's what we are trying to go after. So what we did was that we, being in the hospital, we can do this fairly quickly. So I got 2 more minutes. So 2 studies, one in collaboration with CAMH, another one at TG itself. This is a pain study. So what we asked the question is that, so you see that this is PTSD, that's pain, Albert Wong at CAMH. We have a lot of patients coming in already who are consuming cannabis. So why don't we collect cannabis samples from them and do all kinds of interviews, their blood, everything, so we can bring our chemistry, what's in their blood, what's the clinical diagnosis, all of that together. I'm going to share with you the importance of the substance now as we go forward. So you already know that heating generates the active compounds. So if I were anyone knowing healthcare, if I'm consuming a medicine, I want 100% of my compound to be in this form, okay? That is without those A's in them, because that's the active substance. And when I take my pill or the capsule or cannabis, I know I have 100% in my body. Many of these patients consume something called cannabis oils. And what they do is that they take the oil, they put it under their tongue, or they put it on salad and eat. And what we did was we took those oils from these patients from the clinical studies and tried to look at the chemistry on that. You see two colors there, one is blue, other one is green. And the blue is THCA, inactive compound. Green is the active compound. What you're noticing here is that there is inactive compound, about 10%, 10% here, as much as 80% inactive compound. And here it's all 100%. So these are all different patient samples. All of them are pain patients. So imagine pain patients consuming different varieties of this, right? And this is CBD, CBDA. Again, you see the variability as much as 100% inactive compound in that. And these are receptor assays. I'll shortly here, the idea is that the red ones are inactive in the receptor assays, CB1, CB2. The greens are the active compounds. There's a nanomolar potency with THC, and that's a micromolar potency with CBD. So take-home message out of this is that today, if you take any of the patients that are out there, they have cannabis oil today, they are consuming it, they see effect. A week from now, they buy a new bottle, which is not going to be the same, as you saw the heterogeneity already. It could be totally different, so that it's all over the map as it is today in the industry. We're going to get this in the next couple of months into publication, but that's the bottom line on that. So my goal here was to connect this chemistry and the plant and the cannabis and the patient and the impact of this in the context of medicine. Now, is anybody looking into that in the news that you're hearing about all of that? Not really. Okay, very few people are really, science really hasn't kicked into this field at all. Okay, so hopefully I was able to take you through some interesting stuff happening in our lab, maybe from the science point of view, a few things that you could learn, but also what else would be involved if you're looking at it from the drug standpoint of view. And all the points that you see on the right-hand side, they are very, very important. So as you get into your careers, make sure that those are all the things that you carry and more. This is my last slide. This is one of the 17 stores, something called Fire and Flower in Alberta, as the private cannabis sales point. That's better than Apple Store, if you will, right? With that, I stop. Thank you very much for your attention.
Thank you very much. We're running short of time, so I think we're going to save the questions until later. I'm sure Dr. Kotra will be happy to entertain your questions, and we'll just move on to the next presenter. Thank you very much. Okay, and our last presenter of the day. Do you have a computer here, or he just?
These are my slides. They're right here.
Okay. Okay. Super. Dr. Sergides, Lena. Yeah. Last name?
Sergides.
Okay, excellent. So she is